Hi, I'm Dr. Leslie Blankenship-Williams, and I'm going to be walking you through this genetic engineering lab. This is Laboratory 16 in your lab manual. Now, hopefully in lecture you've already learned that bacteria have a unique capacity to do what is called lateral gene transfer. And what that means is that bacteria can take up DNA and modify their own genetic code to become recombinants. There are three major avenues by which bacteria can do this, including conjugation, transformation, and transduction. In this lab, we are going to try to compel E. coli to transform by taking up a piece of DNA known as a plasmid. Now, plasmids are going to be extra chromosomal pieces of DNA that are small circles. So they're not actually part of the chromosome. They're separate. And the plasmids that we are going to be giving E. coli in today's lab is going to be a plasmid that was genetically created at UCSD. And these plasmids only contain a couple genes, but one of those genes codes for a fluorescent protein. So, if our experiment is successful and E. coli cells do in fact take up some of this plasmid, then we should see them expressing the DNA on the plasmids, which would be for a fluorescent protein, which means they're going to be producing a colorful pigment. When you get to take a look at the results of today's transformation lab, hopefully you will see brightly colored colonies. We actually have 6 different plasmids, each containing a unique gene. The genes for the fluorescent proteins are as follows. We have one for blue, we have one for yellow, we have a green one, and then we have a tangerine one that's called purple, but it's really more of a pink, and then one that's called cherry, which is really more of a magenta purple. So we have 6 different colors that hopefully when you come in, you'll be able to see on the plates. Okay, what we're going to do at this point is take a look at the theory behind this genetic engineering lab. In other words, what we're supposed to be doing and how it works. Now, in order for a bacterial cell to take in a new piece of DNA from the surrounding fluid, that DNA is going to somehow have to be able to cross the cell envelope. This is no small feat. In a gram-negative bacterium, the DNA would have to cross the outer membrane, the peptidoglycan, and the plasma membrane. So how in the world does a large piece of DNA do this? Well, we're not exactly sure, to be honest, but one hypothesis is that there are probably some breaks in the peptidoglycan, and under certain conditions, maybe the bacterium can somehow meld the outer membrane and the plasma membrane together. If you could meld these together, then you would actually create a pore. And that pore might be large enough to let that DNA, as long as the DNA wasn't too big, slip into the cell. Now, it turns out that in the natural environment, bacteria are not generally willing to make pores in their cell envelope because it would subject them to lysis, which would kill them. So bacteria without those pores are of course not going to be able to take up new DNA and therefore will not transform. So we actually have a name that we give to bacteria that are capable of transformation, and the name that we give is competency. So we say that bacteria that are ready and willing to take up new DNA from the surrounding environment are competent. Now, although it is difficult to find competent bacteria in the natural environment, it's actually not that hard to do it in a laboratory setting. And so, in order to make cells competent in the lab, we start with fresh culture, and then we basically add ice-cold calcium chloride. And for whatever reason, as long as we keep those cells on ice with that calcium chloride, they remain in a competent state, therefore ready and willing to take up new DNA. The lab manual has more information on how calcium chloride might assist in making competent cells. Now, in the laboratory procedure that I'm going to follow, which is a little different than the one that's in your actual lab, the competent cells are actually provided for us by our laboratory technicians. So we start with competent E. coli on ice. Those competent cells are provided to us along with plasmid extract that we're going to use for transformation. So to start this experiment, we are given 2 vials of competent E. coli stored on ice. To one, we are going to add 5 microliters of plasmid extract. This is the one that we hope to create transformation with, so we label it T for transformed. To the other tube, we are going to add 5 microliters of pure water. This is going to be untransformed bacteria, so we label it C for control. Here you can see a high-speed video where I use a micropipette to withdraw 5 microliters, which is a very small amount, of plasmid, and mix it in with my competent E. coli on ice. At this point, we let our 2 tubes sit on ice for 20 minutes. The reason why we do this is to let the plasmid mix thoroughly with the bacterial cells. Then we are going to take our tubes over to a 42-degree water bath and immerse them for exactly 50 seconds. This is known as the heat shock step, and if it's successful, this is where transformation happens. Okay, we have waited 20 minutes for our plasmids to mix in with the bacteria. What I'm going to do now is the heat shock step. In the heat shock step, I'm going to take my entire bucket of ice with the bacteria. Hopefully the plasmid is kind of wrapped up pretty close to the cell envelope. And I'm going to hold these tubes in a 42-degree water bath for exactly 50 seconds and then remove them immediately and put them back on ice. During that heat shock step, we hope that the cell envelope starts getting fluid enough through the heat that temporary gaps open wide enough that the plasmid is able to actually make it through both the outer membrane and the plasma membrane of the cell wall and get inside the cell where it will be retained by the bacterium, and therefore that bacterium will be transformed. All right, I'll be back. The heat shock step occurs off camera. Once the 50 seconds are up, the cultures are immediately put back onto ice to cool them off rapidly and stop any transformation. Next is to let the cells heal and then plate them to see what transformed. Let's watch. Okay, that was stressful for the bacteria. They do not like that heat shock step where they have to open up their cell envelope and potentially bring in new DNA. It's very traumatic and could cause them injury. So it's time to give them some nice, beautiful 5-star hotel treatment with some LB broth. LB broth is a lot like nutrient broth, but it's got some extra added stuff that makes the bacteria especially happy. So what I'm going to do is transfer a little bit of the LB broth to my bacterial cultures, and then I'm going to let them incubate at their favorite temperature, which is 37 degrees Celsius, our body temperature, for about an hour in these conditions so that they can heal. And if they have successfully transformed, which many of them did not, but if they have successfully transformed, they'll be able to start expressing the proteins. And that includes the enzyme beta-lactamase that would help cut up ampicillin, as well as the fluorescent protein. All right. Now the reason why we incubate these tubes at 37 degrees for an hour is to give the transformed bacteria an opportunity to express all of the genes that are on the plasmid. Now let's actually talk about the plasmid for a little bit. Plasmids are expressed in an all or nothing fashion. That is, you either express all of the genes or you express none of them. So all of the plasmids that we use come with two genes of importance. One is for the fluorescent protein, but another is for ampicillin resistance. And the one for ampicillin resistance, of course, codes for beta-lactamase, which would cut ampicillin. Now, why do we include a gene for ampicillin resistance? E. coli are not naturally resistant to ampicillin. So by including a gene for ampicillin resistance in our plasmid, it gives us a way to separate the bacteria that have transformed from those that remain untransformed. In this way, it is a selection tool that allows us to basically determine which microbes successfully transformed. Now, how does this work? Well, when we have untransformed bacteria, if we plate them on generic all-purpose media, they would grow. But if we plate them on media with ampicillin, they would die, because ampicillin would destroy the peptidoglycan and therefore the cells would lyse. But the transformed bacteria, those that have successfully taken up the plasmid, now have a gene for ampicillin resistance. So they would still, of course, survive on generic LB agar, but now, because they have beta-lactamase that they've been expressing for the last hour in the 37-degree incubator, if we plate them on an LB plus ampicillin plate, they would survive, because they would cut the ampicillin and therefore be able to grow and produce colonies. So during that 1 hour incubation period at 37 degrees, the bacteria that have been transformed have been expressing beta-lactamase. And so what I'm going to do is go get those cultures out of the water bath, and then we are going to plate them in a procedure that you probably have not done before. So let me go get them. All right, we are ready to begin adding our bacterial cultures to our plates. Now in front of me I actually have 4 plates. I have 2 that are just LB and 2 that are LB plus ampicillin. The A stands for ampicillin, which is an antibiotic in the penicillin family. Now E. coli will be killed off by ampicillin unless it possesses a gene that makes it resistant to ampicillin. Naturally, E. coli don't usually have that gene, which again codes for beta-lactamase, which cuts the ampicillin molecule and therefore deactivates it. But we included that beta-lactamase gene in the plasmid. So if bacteria have successfully transformed, then they should be able to cut up any ampicillin that is embedded in my LB plus A plates, and therefore survive and grow. But if they did not successfully transform, they should be killed off by the ampicillin. So in this way, we are going to be able to select for those bacterial cells that have successfully transformed by killing off all of the untransformed ones. Now I want to talk for a moment about adding in the additional plates as controls. So it turns out that this experiment, like many complicated scientific experiments, often goes awry and produces unexpected results. But if you've included appropriate controls as part of your procedure, it will help you determine what might have happened. So for instance, one semester we had no transformation at all. And so we were actually able to use our controls to pinpoint what happened in that experiment. And in that case, it was that the cells that we got were not competent and therefore did not transform. So the controls that are going to be included are both negative and positive controls. Negative controls produce an expected negative result. In our case, this is going to be cases where we get 0 growth on our plates. Positive controls produce an expected positive result. In our case, a positive control is going to be one where we have lots and lots and lots of growth. In other words, none of the bacteria were killed off. And there are advantages to including both negative and positive controls when possible, like here. So our negative control is going to be the LB plus amp with the control tube, which again is just bacteria and water. So this should produce the expected negative result, which is no growth. Why should there be no growth on that plate? Well, there's ampicillin in this plate, and my control tube has bacteria that are untransformed. So therefore, they should not be growing on a plate with ampicillin. If they do grow, that tells us that our E. coli cultures were already resistant to ampicillin, and therefore this was not a good antibiotic to use as a selection tool. Or if we get a little bit of growth, it could be contaminants, which tell us again that our procedure needs a little bit of work to be aseptic. Now what about the positive controls? Well, the positive controls are going to be just on LB in general. So the positive controls should produce so much bacteria that they grow in together and create what is called a lawn growth, because it actually looks like a fresh lawn of bacteria. So those are going to be lots and lots and lots and lots of bacteria. Now what would happen if our positive controls did not produce any bacteria at all? Well, then maybe we just didn't have viable bacterial cells. Maybe we killed them in the heat shock step. Maybe we did something else along the way. But if we don't see growth on the LB plates, that tells us that something was wrong, either with the LB media or with the bacterial cultures along the way, and it was not viable. Okay, so now that I've explained a little bit about the controls and why we have 4 plates, what we're going to do now is invoke a new technique that is actually really fun. It's one of my favorite techniques out there to culture these bacteria. So let's watch. All right, what I've got is a bottle of 95% ethanol with a glass stick that has curved. Now this is called a glass spreading rod, and what it's going to do is it's actually going to spread the bacteria over the plate, much like you vacuum a room. You just kind of vacuum it. So it's the vacuum technique. It's actually a lot of fun. Now I'm using 95% ethanol because I want to sterilize this glass rod. If I put this glass rod directly in the heat, it would take up all that heat, because glass conducts heat really well, and it would go all the way to my fingers, potentially burn my fingers, and shatter the glass. So this is not like the inoculating loops that you're used to using. So what we do is we dip it in ethanol, and then we run it through a Bunsen burner flame like this. And so what happens is the ethanol burns off really rapidly, so rapidly that it's almost a cool flame. So we've sterilized this part of our spreading rod without getting the rod too hot, shattering it, or burning our hands. Okay, anytime we need to re-sterilize, we simply dip it back into the ethanol. All right, the other thing that we're going to do is we are going to take 50 microliters, again using a micropipette, 50 microliters of our bacterial culture. And then what I'm going to do is I'm going to go ahead and actually just squirt the fluid directly on the Petri dish. I don't know if you can see it. You can see the fluid kind of streaking around there. But in order to spread it out really well, we're going to use our spreading rod. Okay, I am done spreading the bacterial cultures all over the plate. The bacteria settle in and grab on to the agar, and now they are ready to go in the incubator over the weekend, where the transformed bacteria that have produced beta-lactamase should grow on the ampicillin plate. And because they are also expressing the beta-lactamase, they express the fluorescent protein as well. And so when you get a chance to view them, they should be nice and colorful. Thanks for watching.